So if you want to get these slides off of the coursework page, and if you've had trouble doing that, what happens is they give you a menu to pick the application you want to use to open them. But the problem is that PowerPoint, which is what these slides are, PowerPoint is not an option they give you. So I think there's an options button on the bottom left of that menu, and if you click it, there's a drop-down menu that comes up, I believe, that allows you to select all applications. And then it will allow you to select PowerPoint. PowerPoint won't be grayed out. And once you do that, I would hope your computer would remember that PowerPoint slides are opened with PowerPoint. But your computer may not be that smart. So who knows? If you ever get into a jam where you just like your computer just won't open them, just shoot me an email. I can just send you the slides. That works as well, or share them with you, and I would hope your computer can figure it out then, or let me help you with it at office hours, or go to IT. So, with that being said, your homework tonight is to read over the sections that we're going to actually talk about today. So all of this is what you're going to see. And I prefer to be in that position where I talk about it and then you read about it versus reading and then listening to the lecture. So hopefully we can stay on this course. The next test is, I believe, October 7th, which is a Wednesday, which I think is also a day 4. So keep that in mind. That is on the distant horizon. So a lot of the things we talked about in the first unit will start popping back up, honestly, throughout the whole year, especially in this unit. But this unit, to me, I think if I were a student again, would be easier to grasp than the unit that we just got done with. You've probably seen some of these words before, or more of them than you did with the terminology from Unit 1, but maybe not. It just depends on whether or not you remember the organelle song from 7th grade, or if you've had biology before. This first slide is pretty simple. Cells, they're made of organelles. I don't remember the rest. This first slide is pretty simple. This is one of the few times you'll get a diagram that's almost childishly simple in AP Biology, but I really like it because of its simplicity. This is a cell, okay? A cell is just a bubble of like lipid. It's like a fat bubble. In animals, and you are an animal, you're in that kingdom of classification. In animals, cells do not have a cell wall, and we'll talk about that again. The outer membrane of an animal cell, of your cells, is going to be what we call the plasma membrane or the cell membrane. That is the outer layer
of an animal cell.
Of an animal cell. Very good to make that distinction. So I like this picture because it shows the stark reality that a lot of cells face, which is contents, an internal volume here that is full of different things that we spend this whole unit talking about, all protected and isolated from the environment around them by a very thin, squishy barrier. This plasma membrane is mostly made up of fatty molecules, of lipids. And if you imagine like the fat or grease in food, it's not some super strong, solid structure. Okay? So your cells are just these fatty blobs. You are made up of trillions of them. And we will get the microscopes out and you will look at your own cells that you get from your own cheek scrapings, which we will also, you'll do that to yourself, and then you will look at yourself on a cellular level under the microscopes. And some of you may have done that before. Now, the outer membrane here, or the outer layer, is what we call a plasma or a cell membrane, and it's incredibly important. It's what separates you from the rest of the universe in a lot of ways. And this membrane is what we call a selective barrier. Sometimes there's a term, selective permeability, that your book will talk about, and it is referring to the same concept. What does it mean to be a selective barrier? That's right. The inputs and the outputs are controlled. You only let certain things in and only let certain things out. That's the idea of a selective barrier. You are selecting what comes in and goes out of the cell. Now, you aren't doing it, your cells are. But you are your cells. So it's kind of you, if you think about it. Okay. Now, your book talks about kind of the size of these objects and types of microscopes we can use to observe objects of that size. You do not have to memorize any measurements in the centimeter, millimeter, micrometer, nanometer range. I will never ask you what size something is. I will tell you what size something is, and that could matter. And that's kind of what our activity today will look into. Now, if you look at the lines that traverse across this diagram, what you notice is that they are linked up to a measurement with the base unit of meters. And there is a 10 times or tenfold difference between each of these lines. So just like the pH scale, if you go 2 lines, how many times different is that? 10. Each line is 10, so 2 lines is 100 times, 3 lines is 1,000 times, okay? So the scale here, just because 2 things look like they're kind of close, that's a 10 times difference, or maybe a little bit more or less. Okay, so most of what we talk about is here in this yellow range. These are going to be, as it says here, most of your plant and animal cells. They range somewhere between 1 and 100 micrometers, micrometer, but we say micrometer. And that's what that symbol here is representing, the micrometer, which means what? What's that prefix mean? Centi means 100 from century, milli means a million, milli means 1,000 from millennium, and then micro references what? 10,000? Not 10,000, even more.
10 to the negative 6.
It's 10 to the negative 6, which is a millionth of a meter. So if you took this and chopped it up 1,000,000 times evenly, each of those would be 1 micrometer, which is an insanely small amount of length. But that's where kind of the diameter of a lot of the cells we talk about lie. And that's in a pretty wide range. It could be 100 times different in size between different types of, say, animals and plant cells. There are a whole other type of cell out there that we will talk about that bacteria fit in, and they are significantly smaller than your typical plant or animal cell. In the year 2020, I've never even discussed these, even though it's a biology class, we a lot of times gloss over viruses, but we will talk a little bit more about viruses this year for obvious reasons. They are very small, way smaller than bacteria even. And then there's going to be, 10 to 100 times smaller than that, some of the molecules that we talked about in the last unit. Now, you can see down to, a fraction of a millimeter, not a tiny fraction, but some fraction of a millimeter, at which point we typically will use light microscopes, which help us in this range to see most types of cells. The light microscopes are what we're going to use. Your book talks a lot about the different types of light microscopes. We are not going to go into the types of microscopes, except to say light microscopes use light, and electron microscopes use beams of electrons. And they are capable of producing an image with good resolution at way smaller sizes. And I will show you images from time to time. Some of them will be from electron microscopes, some will be from light microscopes, and we'll talk about those differences. Okay? So, how do we study parts of the cell? Well, in your book, they talk about this process, which is called cell fractionation. Cell fractionation. If you want to just say like cell frac as an abbreviation, the book has the spelling and all. I could spell on the board, but we'll move on. But essentially, the general idea here is that you can take tissue, which is made up of cells. So when you hear something like muscle tissue, that simply means a bunch of muscle cells working together. So you can take a cell or tissue sample and you can blend it in a blender. And you can chop it up into all its cell parts. And then what we use is called a centrifuge to spin that chopped up mixture of cells. And when you spin these samples, the heavier stuff tends to be pulled down towards the bottom. So they have this series of spins that they go through here in this diagram. And they remove larger cell parts from the bottom. They spin this mixture again, and you can get other cell parts that are a little bit smaller. Remove them, spin what's left, get even smaller and smaller cell components. So this is a lab technique. We will not do this because honestly we don't have the equipment. No high school really has the equipment to do anything with these smaller cell parts. But this is in general how we get at these parts of the cells that we're going to spend this unit talking about. Now, what I really want us to focus on today are things like this. There are two terms that you must know when it comes to studying cells. Let me get this real quick. And I don't know if you're familiar with them, but what are the two kind of major groups that cells can fall into? Are you familiar with these terms?
Prokaryotic and eukaryotic.
That's right, very good. So the first term I want you to write down is this term, prokaryotic cells. Prokaryotic cells. Prokaryotic cells. Sometimes we just refer to them as prokaryotes, which would be, instead of the IC ending, just an E at the end, a prokaryote. This is an example of a prokaryote. Now there's kind of a list of characteristics I want you to write down about these. So you can just make little bullet points as we go through these. But when it comes to these prokaryotic cells, which is what we're looking at in this diagram, they tend to be very small. These are small cells. They are simple. For a typical cell, they're not very complicated. There's not as much going on as there are in the other groups of cells. So small and simple. There is no nucleus, which is a structure we will talk more about. So if you're not very familiar with that, it's okay right now. What is the nucleus of a cell? When you have a cell with a nucleus, what are we referring to? The what?
Where the DNA is under control.
That's right. It is this. It's a centrally located structure that holds the DNA. That is what the nucleus is when a cell has a nucleus. Now in this diagram, there is no nucleus depicted because prokaryotic cells don't have a nucleus. That's your microphone. Thank you. Now there is this region where you find the DNA typically in a prokaryote called the nucleoid. You may see that term referenced, but it's not a nucleus, and I would never ask you to define it. And along the lines of it being simple, these prokaryotic cells being a simple type of cell, that not having a nucleus or there being no nucleus, that's one part of that concept of this being a simple cell. But they also lack a number of other organelles. And that's another term you may or may not have heard before. This term, organelles. These are basically what we would call cell parts. You've heard of some of them. Name me some organelles. Mitochondria. That's the one everybody's heard of. I think that's what you were saying back there, Zoe. Mitochondria, the endoplasmic reticulum, cytoplasm is sometimes referred to as an organelle, even though it's more of just a fluid than a structure. But organelles, which are cell parts, are not missing, but there aren't a lot of them in prokaryotic cells. So a lot of times what I would say about a prokaryotic cell in terms of a bullet point description of them is they are lacking. They're not absent, they're just kind of lacking in organelles. And typically, all you're really going to find are these ribosomes. So they only really have ribosomes and maybe a few other structures. Now again, the beginning of any term gets a little bit weird because in the next week or two, you're going to know a lot about ribosomes, even though you, at this point, may not have heard of them, but we'll come back to some of these terms. The last thing I want you to write down about prokaryotes before we move on is this word right here. This is what or how you would describe bacteria. Now, this term bacterium is the singular. Bacteria is actually plural for many of them. But this is an actual electron microscope image of a specific bacterium. It is a prokaryote. It is very small. It is very simple in that it doesn't have a nucleus. It has ribosomes, but not many other organelles. A lot of times if you say the word germ, germs, that kind of common everyday term, that is usually a reference to the bad prokaryotes that are out there. But there are a lot of good ones that live inside of you, help you digest food, actually attack bad disease-causing prokaryotes. So they're not all bad. Don't think of bacteria as a bad word. All right. The other type of cell is what?
Eukaryotic.
That's right. Eukaryotic. Eukaryotic cells. So we're going to do the same thing with the eukaryotes. Now this is only a micrograph, not a diagram going along with it, but I will show you a diagram. This is from a light microscope. We will see this in the lab. What you're looking at here is a eukaryotic cell. And the first thing I want to point out, actually I want to go back real quick. Whoops. Actual image, about a half of a micrometer in width here. Okay? 0.5 micrometers. Versus this eukaryotic cell, that's 50 of the same unit, as opposed to 0.5. So instead of a half a micrometer, this is, and that's only that distance at 50, this is probably about 100 micrometers wide, or 100 micrometers in diameter. So this cell is 200 times bigger, roughly, than the cell we saw in the previous slide. So under eukaryotic cells, you definitely want to indicate these are larger cells. And what else? They have a nucleus. That's what you're looking at right here. That is the nucleus of an eukaryotic cell. You also see all these other kind of dots and whatever shown around the nucleus. Those are a wide variety of organelles. So this is a much more complex type of cell. So when you compare eukaryotic to prokaryotic cells, eukaryotic cells are bigger, more complex. Having a nucleus is a really key difference or distinction between the two. And you are eukaryotic. So plants and animal cells are eukaryotic cells. If you can see it and it's alive, it's eukaryotic. And when I say if you could see it, I mean without a microscope. Anything crawling around in nature that is alive, that you can see with your unaided eye, is a eukaryote. One other distinction that I did not make about prokaryotes, so you might want to add this to the other one, but we'll start with eukaryotes. Eukaryotic cells, these can actually be multicellular organisms. I mentioned you were made of trillions of cells. Only eukaryotic cells have the complexity to work together in large groups. What does multicellular mean? Many cells, multiple cells, that's all that means. So eukaryotic cells can appear in multicellular, but with prokaryotic cells, that is a single cell acting alone. Only single-celled organisms make up this group known as the prokaryotes. You can have a single-cell eukaryote. They're not uncommon. If you've ever heard the term amoeba, that is a single eukaryotic cell acting alone by itself. But in general, eukaryotic cells are the type of cell that can make a multicellular organism. Okay, so just a couple more slides here. No matter what kind of cell you have, prokaryote or eukaryote, they're going to have a cell membrane. We looked at this a little bit last unit. Your book referenced it a little bit last unit. And there are some things, name me some terms from last unit that you start to see up here. Yeah, here we have our good buddy, the phospholipid, with the head and the tails. And as it mentions here, the head is the hydrophilic part, and the tails that are just long fatty acid tails and are nonpolar are hydrophobic, and they don't interact or like water. And so you get what we call a bilayer. You get these two layers of phospholipids where the tails are pointing away from the water outside and the water inside the cell. And the heads are pointing towards the water that is outside and towards the water that is inside the cells. So it's a really natural kind of structure for these phospholipids to make. And all living things take advantage of this. Every living thing has a membrane that looks something like this. Viruses, like COVID-19, do not have a cell membrane. And guess what? It's not a living thing. It's just this weird machine that takes over your cells and destroys them. Viruses are weird. Up here, this is from an electron microscope. This is basically what you're looking at, except a real image of this diagram. Now, there's more going on than just the phospholipids. There are proteins and these carbohydrate side chains that are referenced here. And we're going to get into a lot of depth of the cell membrane. But I want you to keep this in mind, the cell membrane. I want to say one more thing about the cell membrane, and then we're going to talk about why that thing is important. The cell membrane, as we already talked about, is what controls inputs and outputs for the cell, okay? So just like you, and you're just a bunch of cells, so there's a good reason you act the same way they do, because that's what you are, is a conglomeration of these cells. Just like you, every cell needs, oxygen and food brought in. They eat and breathe kind of, not like we do, but in that same manner of inputs like oxygen and food are important. They create waste products like carbon dioxide and other chemicals that can no longer be used, that can be toxic if they build up in high quantities. So they got to bring in nutrients and get rid of wastes. And that is the job of the cell membrane. Remember that. This is the only part of the cell membrane, this outer layer, that is the only part of the cell that can get stuff the cell needs in and kick out waste products. So that carries us to this next slide. So keep that in your head. The cell membrane, which is the surface of your cell, of all your cells, is what brings in the good stuff and gets rid of the bad stuff. That's the part of the cell that does that.
So is that the selective barrier?
And that's part of the selective barrier characteristic of the cell membrane. It selects what comes in and out. Obviously, it wants good stuff and it wants to get rid of bad stuff, to put it simply. Now, this is where a little math comes in, and this is what our activity today is going to be about, and we'll get started on it in about 5 minutes. Surface area and volume. These are concepts you learned about in geometry, right? Surface area and volume. You had to calculate it for different shapes. What shape did I say an animal cell is?
A circle.
It's spherical. It's a three-dimensional circle, a spherical shape. Now, in this diagram, we're using cubes, but that's kind of the shape of a plant cell. Plant cells are kind of blocky in shape. So it doesn't matter what shape you use. Cells come in a lot of shapes, but animal cells do typically have that rounder appearance. Now, what this chart is showing you, especially this bottom, and I'm really looking at these two columns right here, but this bottom row is showing you a calculation of surface compared to volume. Now, I want you to think about that. Surface is just, if you were to look at a cell, is only what part of the cell? The surface. The outer layer or the cell membrane. So when we talk about these cells of different shapes, the cell membrane is your surface area, and your volume is what? All the rest of it, the entire insides of the cell. That's right. So when you say, and the AP exam loves stuff like this, taking mathematical models and putting them with biological concepts. When you just say surface area to volume ratio, which is very much a mathematical term where you simply take surface area and divide it by volume, that ratio is, in real life for cells, how much cell membrane you have versus all the stuff inside the cell that needs nutrients and are producing wastes. Okay? The more volume a cell has, meaning the bigger the cell is, the more needs it has. It needs more oxygen. It needs more food. It's making more waste to get rid of. And what is responsible for dealing with all those needs? The membrane. So look at what happens. We have a tiny cell or shape versus a big cell. And they don't have units up here. They just call these the units units. So this cell here is 1 unit along the side of the cube. This cell is 5 units. As we go from a small cell to a big cell, look at our ratio of surface area and volume. It drops pretty dramatically. Okay? So the bigger a cell is, the less surface it has compared to volume. What's the problem with not having enough membrane for a big cell? It's not getting the food it needs to get or the oxygen it needs to get, and it's not capable of removing the waste that it's producing. Bigger cells are less efficient. It's why, thankfully, you don't go outside and just see a giant building-sized cell just coming at you to engulf you and digest you. They can't get big. Honestly, for pretty much the same reason insects aren't as big as they used to be, because they can't diffuse in enough oxygen like they could 150 million years ago, because there's less oxygen today. So this concept, you'll be beaten with this concept over and over. Small cells more efficient, big cells less efficient, and the reason is smaller cells basically have more surface to deal with the needs of the insides than the big cells do. And finally, I'm going to spend less than 1 minute on these next 2 slides. Your reading goes ahead and jumps into animal cells, which you see here. This is a diagram of an animal cell and the diagram of a plant cell. One challenge of this unit, and it's not a bad challenge. It's not too intimidating, I don't think. It might be at first, but we'll go through all of these terms over the next few weeks. All of these cell parts or organelles are things that you need to be familiar with. Animal cells have a slightly different list than the organelle list you find with plant cells. And those are the two major types of eukaryotic cells we will deal with. Now, again, you're reading and in class, we are going to go through all of these parts, or almost all of them. So you're going to need to get familiar with these names. And that's, again, just kind of part of the vocabulary of this unit. To help with what we just talked about in terms of review, I'm going to hand this out. This is also on your coursework page under Task C, Lab, Cell Size and Organelles. And it will count as a lab grade. We get started now, and we will have time next class to continue working on it because we only have about 20 minutes in class right now. If you could stop the video, and Tanel, if you